This lecture will be looking at and describing some of the major trends we see in animal form, and some of these were just re-emphasizing, maybe giving a term, a defined term to something we've already talked about. Now, when I say major trend, I'm not talking about, like, for instance, how are snakes different than lizards? I'm talking bigger picture here. We will, in some instances, look at some of those, I guess, more minor, it's still major, but less major trends. But I'm talking big picture across all animals. What are things we see that we could apply to just about all animal groups? Certainly all bilateria, these large, complex animals. And so we're going to look at four really big ones that really are kind of universal across the major groups of animals. Okay, the first one we've already talked about, and this is this idea of modularity, that we can increase and decrease the number of repeated units. Now, the technical term for this, which we haven't given you yet, is meristic variation. So meristic variation is just changes in the number of modules we have in an organism. And we can see lots of great examples of this. For example, the number of vertebrae across the chordates varies widely. Fish sometimes have dozens of different vertebrae. Snakes, sometimes we get close to 100 or more vertebrae. There are variations even amongst more conserved groups. So, for instance, although most mammals have 7 cervical vertebrae, there are a couple of sloth species that have an extra cervical vertebra. So we can see this and realize that these types of mutations are very, very common. We can see that sometimes even in the population diversity, where we'll have some species that might have a varying number of segments. Usually, though, it's fairly conserved within species. If there's a mutation, it's either advantageous or disadvantageous and will either spread through the entire species and become the ground plan for that species, or it will be removed because it's a detrimental mutation. But these meristic mutations that change the number of repeated segments is very, very common. Teeth are another good example. And this is one where there's more intraspecific variation amongst mammals, even among humans, right? Where we have, it's quite common for individuals and humans to have supernumerary teeth, right? Extra sets of teeth or just an extra wisdom tooth or something like that. So that one, there's a little bit more flexibility. Whereas with number of vertebrae, although there are some mutations, those are less common and have a bigger, usually detrimental, phenotypic impact. Okay, so just know the definition of meristic variation, and it's one of the major trends that we see across all animal groups. We've already talked about this diversification of these serially homologous parts. So just remember our definition of serially homologous. It's a repeated module that originally, at least for a short period of time, was very, very similar, maybe even identical to the one anterior and the one posterior, or the one proximal versus distal. We'll look at axes of animals. If you're not familiar with those words yet, that's okay. But a repeated structure like a segment in an arthropod that originally was ancestrally was identical or very, very similar, that then can go on to diversify. And so that diversification leads to many, many different functions while still having the possibility of maintaining the ancestral function for those segments in at least some of the serially homologous units. Okay, now in addition to diversification of serially homologous parts, we can also look at and begin to define and investigate the diversification of homologous parts. So when we say just homologous without the serially in front of it, we mean organisms that can be traced back to the exact same structure in a common ancestor. So for instance, the forelimb of all vertebrates has diversified widely. So I don't know, I repeat this one here, I guess just an accent there, but the frog, the lizard, and the bird have different forelimbs. I could put a human and a lizard, I guess we already got a lizard, sorry, a human and maybe a bear, a cat, and we would see differences, although enough similarity to establish that they are homologous to one another just on the basis of morphology. But with enough differentiation, there's some really interesting questions to be addressed and evolutionary changes to try and understand. Okay, so one of which we've already talked about is this kind of ground plan of all tetrapod vertebrates have, that are living at least, some of the extinct ones had more, but all living ones have a ground plan of 5 digits, although some, like the bird, have reduced those digits. So birds actually do have phalanges, digits, but they've been greatly reduced. They have 3 instead of 5, although their ancestors had 5. Okay? So that diversification of homologous parts, and don't get that confused with the diversification of serially homologous parts. So if I was comparing the forelimb of a bird to the hind limb of a bird, I would be comparing serially homologous structures, or the forelimb of a bat to the hind limb of a bird. Again, that's serially homologous structures. If, however, I compare the forelimb of a bird to the forelimb of a human to the forelimb of a lizard, that's looking at and comparing homologous structures. So you can see why these definitions are important. It gives us a frame of reference upon which we can begin to build and gather data to answer questions about how these organisms evolved. All right, the next major trend that we see is, of course, the evolution of novelties. Now, defining novelty actually is a little bit trickier than you might think. And I'm going to give you 2 definitions, but I want you to realize that when someone says novelty, you need to be a little bit careful because there's a little bit of a range of things that they can meet. So let me give you some example of things that we might consider novelties. Teeth, insect wings, a feather, and there are many, many other examples. Now, I chose these because for a long period of time, and to some extent, especially for the insect wing even today, there is still a big mystery as to what was the original structure that gave rise to these things. So, a very strict definition of a novelty is a brand new structure that has an important function. And what I mean by a brand new structure, that there is no clear evolutionary ancestor of that structure. So for instance, for many, many years, we did not know what the evolutionary ancestor of a feather was. What did it come from? And in fact, it took us until Evo Devo and looking at the genetic underlines of how we build a feather to realize that feathers were actually built from a somewhat similar pattern as reptile scales. The genes that build feathers are in some ways similar to the sets of genes that build scales. And so that gave us a connection. We realized that we know now what the evolutionary predecessor of a feather was. Now, does that mean it's not a novelty? Well, most people would say, no, it's still a novelty, but now we understand its origin a little bit better. So that gives us a little bit of a looser definition of a novelty. A novelty, in addition to being a new structure with an important function, it can be an old structure that changes enough to gain a new feature. So maybe it's the function of that structure that's a novelty and not so much the structure itself. So wings, for instance, in birds are a very good example of that looser definition of a novelty. We know exactly what the evolutionary predecessor of the wing was, right? We know it was a forelimb. We don't know all of the steps along the way, but many of them we can see in the fossil record. But we don't have to wonder or try to figure out, oh, where did this structure came from? We know that it's a modified forelimb. So it's still a novel feature because it's doing something that its ancestors couldn't do. But the structure itself, we clearly understand where it came from. Now, with insect wings, we'll look at that later. I don't want to spoil it for you. You can look it up if you're interested in. But insect wings, to a larger extent, for a longer period of time, remained a little bit more of this stricter definition of a novelty where not only is it a brand new function for that organism, but we didn't know exactly where they came from, what evolutionary ancestral structure gave rise to the wings, because in the fossils, the story was not as clear as it has been for the feather, for instance, or for bird wings, maybe is an even better example. Teeth also. Teeth are certainly a novelty, and their genes help us understand, but we look back in the early chordates, placoderms, ostracoderms, and we don't have teeth. We have jaws. Jaws develop first, but we don't have teeth, and it's only later we get these repeated structures to develop into teeth in each individual repeated unit. So teeth are a really good example of kind of the stricter definition of novelty, that they appear in the fossil record and there's no clear evolutionary predecessor to those structures. Okay, so just know what a novelty is and know those kind of different ranges, a very strict definition and then a little bit of a looser definition. And then we're going to spend the rest of the semester looking at some specific examples, but also general trends that we see when we look at DNA. So how do we go from a single undifferentiated fertilized egg to a fully formed organism with hundreds, at least dozens in animals, but in most animals, hundreds or several hundred different unique tissue types, those tissues organized into organs, those organs organized into different repeated segments or modules. How do we get from this simple structure to this more complex structure? And again, DNA, the genes, that's the link, right? At the genetic level, there is, albeit very, very complex, but there is a process that basically takes us through this idea of novel morphology being developed. So in very general terms, the answer to Aristotle's idea about formation, remember he called that epigenesis, going from no form, and he didn't have microscopes, so he couldn't even see the cellular level. And so he would say going from no form at all to a complex form. Maybe today we'd say going from a very simple unicellular form to complex form. That is epigenesis. And the answer in general terms to how that occurs is genes or DNA. But we're not going to be satisfied with just saying, oh, the DNA does it. We want to know exactly how. We want to begin to investigate and study the details of how those strings of molecules determine and dictate the form that will be made. And we're going to spend the rest of the semester doing that. We'll still refer back for examples and for foundations to the morphological structures and set up these ideas, but we really want to begin to delve into the DNA. And what we'll begin to find is that it's important what you have, but not even as much what genes you have, but how you use those genes. And that's been one of the major kind of take-home messages is that have come about from our study of Evo Devo, that often the same basic modules at the genetic level, the same genes, can be used and repurposed in different ways so that with the same basic toolkit, you can have a different outcome by simply using those genes and connecting those genes differently so that they have different action on the early embryo.